wanted to welcome also everyone and thank you for taking the time to attend this meeting. I mean, we've been working on polymers for quite some years now, and we thought that it would be helpful. I'm not gonna use a lot of time. I'm gonna pass the floor to Ellie Onagil, that is a member of the ECETOC Scientific Committee. She will take you through sort of more details, agenda, and so on. So that without taking more time, I'll just pass the floor to Ellie. I hope you're going to enjoy that meeting today.
Okay, also from my side, a very warm welcome for today's webinar by ECETOC. I hope you can see the slides, everyone. If not, please shout. Okay, I see some nodding on the camera. That's good. So first thing about housekeeping for today. There is a chat box in the webinar which you can use to type your questions on the talks already during the talks. But we basically foresee half an hour of Q&A time to talk about the questions after the talk. So we'll not be responding to questions during the presentations themselves. So either you can take notes of what your questions are for yourself, or you put them into the chat box right away, and then we'll collect them and bring them up in a useful order, basically in the Q&A later. As Andrea has mentioned, the webinar is being recorded, but not the Q&A, and we would like to ask you to keep your microphone muted and also the camera off during the webinar and during the presentations. So the presenter will be on video, but not the others. Okay, and so my intro and Olivier's intro is very short, 2 more minutes to go. And then we'll have 2 parts of a talk by Jens Otter from BASF. He'll talk about the general ECETOC conceptual framework for polymers, especially the grouping approach, which is proposed within that conceptual framework. And then we will look into 3 examples, because certainly you can discuss a lot in theory about certain approaches, but the strengths and weaknesses then will only become apparent when there are examples discussed. After that, a very short break, a bio break in principle, and the Q&A. Okay, and so the question is a bit, what does this mean and what do we want to achieve with a polymer grouping framework, right? As scientists, we basically have 2 asks from society to follow up on. And those are, first of all, reducing animal testing. And the second one, equally important certainly, is to reduce uncertainty in chemical safety assessment, risk assessment. And those 2 objectives could be superficially contradictory, but if we are strategic about it, we can basically define frameworks of how we can achieve the 2 objectives at the same time with a maximum outcome. And the question is a bit, how do we do that from a technical perspective? And the question is, how do you design groups for risk assessment? How do you design what tests you do on the different members of a certain group? Right, when you don't think about how you structure your group up front, but if you start to collect data on different types of products without being strategic about it up front, then your building, so to say, your risk assessment building for such a group or for similar substances, at the end of the day looks like the left Lego figure here. And those 2 figures are built with exactly the same amount of Lego stones, supporting a roof, so to say. So this is more a transformation or a translation from Legos into risk assessment for groups, basically. And the left figure in principle represents your risk assessment building if you use testing which is just based on single substance triggers with a subsequent read across, right? But if you think about what testing you do on which test materials up front for a whole group, then you are much more able to build a more strategic structure, and you're certainly reducing the uncertainty. You're increasing the stability of such a structure, and when you transpose that to a risk assessment, you're reducing the uncertainty in the risk assessment by a strategic selection of your test materials and the study types and the group registration. And with that, we see that there is an opportunity for both aspects, reduced uncertainty and also less animal sacrificed, if we can come up with a good framework for grouping of polymers, right? So the question is, what does such a framework need? It needs to use less animals, it needs to reduce uncertainty in risk assessment, and it also needs to be flexible for different types of chemistries, because certainly polymers are a very broad space of different properties. And equally important is that today we have a webinar with a good discussion and a review of examples and achieving a good understanding of the ECETOC proposal for this polymer grouping approach, because such a framework needs to certainly be discussed with different stakeholders. And we also acknowledge that this is not the end of it. I mean, not the final setup, but certainly there would be more guidance needed to be developed, and there may be some details of such a framework to evolve. And that makes it even more important that you are here today and you get a good idea of what the ECETOC Polymer Task Force has been thinking and what this might mean for specific examples, for example. So we'll first, as mentioned, present the concept with some examples, and then we are looking forward to an open technical discussion on the proposed concept. And with that, I'll just shortly introduce Jens Otter. He's a biologist with a master's degree in ecotoxicology and also PhD ecotoxicologist with more than 10 years of experience in risk assessment of chemicals, working for BASF in Ludwigshafen. And as you can also see by his involvement today, he's involved in several working groups at ECETOC, ICCA, CEFIC, and ACC level. So I'll unshare. I hope that works.
Thank you very much.
Okay.
Thank you very much for the introduction, Heli, and my pleasure here to being here and presenting the overall framework and one of the examples. So welcome to this talk. First of all, I'm going to introduce you and to this conceptual framework for polymers grouping approach of the upcoming report, ECETOC Task Force Report 133-3. So first of all, if we are looking at polymers, we ask ourselves, why do we need such a dedicated grouping approach for polymers? So what is so special? Why do we make our heads around now? And the reason is rather simple as complicated at the same time, because it's the building block nature of polymer chemistry. So related polymers are often homologues manufactured from the same starting materials and similar processes, but finally leading to a large number of similar structures, similar but not definitely the same. And even if some of the building blocks may be different, and such chemical variation in a small part of a macromolecule usually does not lead to differences, to large differences in physical, chemical, or biological properties. And this is the basic thinking of the entire idea of putting polymers into groups. And how have we done this, or how does our proposal look like? Very roughly, we built on internationally agreed grouping concepts, and these have been further advanced to better scope with the first bullet point. So, and a central role that we have assigned into this process is the term hazard similarity. And you may now ask, okay, what does this term mean? And I hope I will bring it up to the table during the next slides and minutes. So this term will be the overarching aim of the polymer grouping approach, to define the similarity of different polymers and consequently a final group that should help us to cover this topic. So the approach proposed here finally should allow to significantly simplify but also structure the data requirements. So as you have seen in the Lego figure, as Heli introduced. So to simplify into structure for polymer hazard and risk assessment, and finally to give a pragmatic and reasonable prescription of substance identity. So coming back a little bit to the first ECETOC report 133-1, which roughly gave the frame for the conceptual framework for polymers. And there we had a chapter on how grouping could be done. And with the current approach, we want to highlight this a little bit more and bring to more details. So in this chapter in grouping, we introduced 5 steps. So step 1 and step 2 should mean to use expert judgment to identify key parameters and determine polymer similarity. And the second and the third step, you define the hypothesis for grouping and you determine the relevant approach. You finally identify the relevant data in step 4 for fate, ecotox, tox. And finally, in step 5, you justify and document what you've done there along all 3 criteria that I will further lay out, and to make this clear and transparent what has been done in all this step and how to justify the group based on the data. So all these steps are not necessarily passed through in the consecutive order. They may be going back a little bit stepwise. However, this is the rough approach. And this approach today, this enhances the outline of steps 4.1 to 4.5 by introducing and setting aside 3 criteria. So a 3 criteria approach set aside to this to define and to justify polymer similarity throughout these 5 steps. So how does these 3 criteria look like? These are brought up here. How do we enhance the grouping approach? And they're basically brought up on the left side. We have 3 criteria. Number 1, chemical nature. Number 2 are the phys-chem properties. And number 3 are the ecological and toxicological properties. And these 3 build the basic principle for polymer grouping. So you follow these 3 criteria and you balance them. Then the aspects included in these criteria are all balanced in this to finally reach similarity towards the hazard. And all these 3 criteria serve for doing so. And they're critical also to form the content and the boundaries of the group, and finally of substance ID, the substance IDs. And substance ID is not another issue which is more or less not really solved for polymers. What is one substance and where does the next one start? And especially criterion 3 is needed as an overarching property. So the ecological and toxicological properties, because this is a fundamental property for chemical registration or regulations globally, because we're not doing registration entirely to describe the chemistry, but we only want to work towards hazard and especially risk assessment. So you need criterion 3 to determine your group. How can this be done? And this is something I'm going to introduce you on the next slide. To provide you with further details on these 3 criteria. How could this work in this schematic graphic? So first of all criteria are indispensable to describe hazard similarity of the group. And while hazard similarity is taken as the central element and the ultimate goal, while defining the polymer group. So how does it work out if you go on the left side of this figure now? You see a hazard similarity is the overall target with the arrow on the left side. You start on the top with criterion 1, chemical similarity, and you take a real bunch of polymers that you would like to group. So the first criterion is now towards chemical similarity and the chemical property. And in this graphic, we took a box and a random property here and sorted them accordingly. Criterion 2 is then the next one to follow on your way towards targeting hazard similarity. So here we sorted again the 2 fields that we identified above. We sorted them for large and small members. This could be a property, for example, for phys-chem. And then in criterion 3 on similarity regarding ecotox and tox properties, we put yellow and blue. So you see that in a stepwise approach, you go down and identify what you finally want to describe as your final group. And once you've done this, And depending on the use types, you can go on with further bracket testing for risk assessment as necessary. And this may, of course, include that you may need to go back and redefine what you've identified before based on new knowledge that may arise. The next slide, I will give you the overall concept. So this is the most complicated figure that I can present to you today. But firstly, I will guide you through this figure because it's a really busy one. So on the left side, you see everything what I already presented. So you see that the 3 criteria should serve for hazard similarity. So you start with your polymers and you work towards hazard similarity. And finally, you should be able to establish and describe what you aimed for. And while doing so, you take criteria 1, 2, and 3 equally into account. If you now move to the middle part of this figure, you see that this polymer grouping approach is a stepwise approach. So you're stepwise narrowing down of a particular group as is described with different iterations. You see iteration 1, iteration 2, and you finally reach up with the final group. But maybe you also need a third iteration. It depends on what you're finally aiming for grouping. So this means you start with the universe of polymers in the green box, you go down your iterations, and finally reach a final group that you're able to describe by chemical nature. So you see different colors, and each color represents one final group. And within this final group and criterion 1, you should be able, by describing the chemistry, to identify the common key feature, a feature that is common to all members of one of these preliminary groups, for example, here in yellow or in light yellow. So there needs to be one clearly defined feature, common key feature to the ones in strong yellow and the light yellow, and the same for all other colors. And the definition of the group is further substantiated by criterion 2 by the PhysChem, by identifying relevant physical-chemical properties, and also by criterion 3, hypothesizing the relevant hazard properties. So which hazard is relevant based on the entire set of hazards that we know about, or the relevant hazards that we could think about. So it's always a matter thinking what is the relevant hazard and what is the common key feature that is linked to this relevant hazard, and to bring this into the definition and thereby define clearly the borders of the group. So you may now ask yourself, okay, what is the relevant hazard? Or what is the way of similarity that should be achieved within one of the groups? And therefore, I want to present you on the next slide and bring some more basis to the level of similarity that should be achieved. So first of all, the effect level of relevant hazard properties of group members from one end of the group do not necessarily have to be the same as those group members at the other end. So this is an important part. So the relevant hazard properties as such, for example, acute oral toxicity, need to be the same for the entire group. But overall, the continuum and predictivity of the relevant hazard property should be established and justified. So this is what's needed. And we tried to plot this into the graphic on the left. So you see on both axes, for example, on the x-axis, a continuum of property related to common key feature 1, whatever key feature 1 is, some chemical property. And then you see somewhat a related effect level of a relevant hazard. And you see the hazard is not the same, but it's predictable. And therefore, there's a continuum, and it's predictable by its effect level. So this is something towards the similarity, and I'm sure the following example will bring this a little bit closer. So how can we visualize all this? Therefore, we want to introduce the data matrix that should help us. A data matrix supports the evaluation of the polymer grouping approach to illustrate the common key feature as well as corresponding relevant hazard properties. Thereby, the consistency and predictability of the relevant hazard properties along the common key feature can be assessed and clearly documented. So for example, on both axes, you can bring up the common key feature, for example, common key feature 1 and a common key feature 2. So let's say, for example, one structural element and the other one is a phys-chem property, or also another structural element. And then within this matrix, there can be a color coding or whatever is needed to illustrate the hazard and the continuity of the hazard within this matrix. So you see it's a shading, but there's no clear border. So something else has to be kept in mind, and this is on the testing on hazard data, because there may be now many members within the group, and how can testing be done in an appropriate way? So to cover the variety of polymers, so-called bracket testing can be applied when additional endpoint data are required to characterize the hazards. And some different test materials out of the group are selected for testing and subsequent read across of the properties across the members. So this requires selecting those test materials which are, for example, at the boundaries of the group, and some members that are more in the middle of the group, and that should be descriptive in terms of chemical nature and phys-chem properties. So which means that we have within one substance could be several tests, which means that the number of test items in the bracket tests necessarily to adequately represent the entire group, and this will depend on the range and consistency of criterion 1 and criterion 2, the already available data within the polymer group, as well as on the concordance of the findings of any bracket testing. How about risk assessment to characterize the final group? So this may also be subject to the entire group. And how could this be done? And again, this is founded on the common key feature and the relevant hazard properties. And again, we took this matrix illustration to make it a little bit clear. So it may be necessary to subdivide the final group to further define the exposure or hazard assessment depending on the margin of safety or on different uses and associated exposure. So there are two examples on the left. So one where the hazard is rather same or similar, or the use is rather same and similar, and the reference value for risk assessment is rather the same. So you could go for one risk assessment. In case where we have a continuity of effect level and where you may need different reference values, you may need to subdivide the entire group into clearly defined subgroups with clear subgroup borders and run 3 risk assessments for parts of the entire group. And as said, this may depend on the margin of safety of different uses and associated exposure. So that's so far. So to summarize on this overall concept, the building block nature and resulting complexity and versatility of polymers chemistry serves a dedicated concept for grouping. The polymer grouping approach described here should be viewed as a targeted and flexible framework. And the internationally agreed grouping concept has been further advanced, and the concept for hazard similarity has been added to cope with the complexity. The description along the 3 criteria also allows the description of polymer groups and polymer identity. So this is another issue that could be solved within this approach. And as usual, I mean, certain steps of the grouping approach may be higher, of lower importance for some types of polymers, and whereas other types of polymers may benefit from adding specific aspects. So this is the overall concept. With this, I will lead over to the first example to illustrate a little bit better what has been presented here more theoretically. So the first example presented today will be on grouping across a continuum of polymers, and this example is along the alkyl etoxylates. The alkyl etoxylates, you brought your schematic presentation on the left side, are surface active, non-ionic polyethers of long chain primary alcohol. You see on the left here, the primary alcohol reacted with ethylene oxide. This is the part on the right here. And these guys are linear, quasi-linear, or branched. So the one on the left is a linear one, but they can also be quasi-linear and branched ones. They're very much commercially available, and for these that are commercially available, the degree of branching as well as the chain length distribution varies by the feedstock source and by the method used to produce the alcohols. So this brings up back to the attention that polymers are a mixture. So there's no single one, but usually if we go for testing of alkyl etoxylates, it's generally a mixture based on the primary alcohol and the length, the chain length of the reacted ethylene oxide. So the use of alkyl etoxylates is more or less towards surfactant use, emulsifier, wetting agents, and moderate formers. And coming to the descriptors that are used so far, so the standard chemical descriptors like CAS are currently not sufficient specific to really identify alkyl etoxylates. So there are a bunch of CAS numbers for the alkyl etoxylates to describe them. However, they're not specific enough to really bring them towards a registration or whatever is needed to define these type of chemistry. So how to apply the overall framework to this chemistry? I will illustrate you on the next slide. So first, step one, we use expert judgment to identify key parameters. And number one is the chemical nature. So they are considered to all have the same structural key element, which is the common key feature. And the structural key element you can see on the left. This is a hydrophobic alkyl moiety and a hydrophilic polyoxyethylene moiety. You see on the right. The general structure is given here. So it's always a certain range of C chain length and etoxylation degree. And we're in that range and defined by these numbers. The molecular weight is given here from approximately 2000 to 2500 Dalton. And although different by C chain and degree of etoxylation, they can be considered similar because they share the same structural key element, which is given here on the left. And one example of on the left downside, you see the homolog distribution of a fatty acid that would be called C8 for EU, which means that the C chain length is at about 8 and the etoxylation degree in average is at about 4. But if you see this, even if it's at 4 in average, there are many, many other species into that mixture. So there is always a mixture, and this mixture is two-dimensional, which means varying in C chain length and etoxylation degree. So step 4.2 is towards the expert judgment to determine polymer similarity, which focuses pretty much on criterion 2, the fifth chemical and fate properties. The common key feature, as introduced on the previous slide, of the linear alkyl etoxylates triggers criterion 2, which means that they reduce the surface tension below a certain threshold and they're rapidly biodegradable. So this determines the borders of the group regarding criterion 2. And to summarize those both steps, alkyl etoxylates are included as defined as being linear, having the general chemistry structure as given here with these numbers, and as reducing the surface tension of water and being readily biodegradable. And adding surface activity and readily biodegradability for defining the borders of the group defines a clear definition of the group. So chemistry itself does not help. We need other identifiers to aid us in defining the group. So step 4.4, I will now make a short switch to step 4.4 to introduce you to the hazard data first before I go back to step 3 and introduce you now to the data. So first of all, the ECETOC data. So for alkyl etoxylates, there is an existing comprehensive data set on acute aquatic tox data. And also there's good abundance of chronic aquatic tox data. So we can really dive into. So what is the background for grouping? What can we build up on the data that is available? This is that aquatic tox for alkyl etoxylate is driven by the non-polar mode of action, resulting in strong structure-dependent increase in aquatic tox, increasing with overall hydrophobicity of alkyl etoxylates. So this brings me to the figure on the right. So if you see that the structure indicates high hydrophobicity, this means also rather toxic effects on the aquatic acute side. And if the hydrophobicity is low, we will see a non-toxic effect. So this means that we do have a continuum. And along this continuum, we can now try to subgroup. We put as example now 3 subgroups, but you can also think of just 2, or you can think of 5. I mean, it's a continuum, and you can build up subgroups and justify within the subgroup. Regarding other ecological endpoints like biodegradation, bioaccumulation, they're mainly determined by the metabolic pathways of the alkyl etoxylates due to the same common key feature, easily biodegraded and are metabolized. Therefore, regardless of the precise structure, they are readily biodegradable and have a low tendency to bioaccumulate. So, as pointed out here, the relevant data are within the acute tox. So we made use of the hazard matrix to illustrate the existing data. And this data matrix includes all commercially available alkyl etoxylates that are included in the CESE recommendation. So this is freely available on the CESE website. You can download and see how harmonized classification and labeling of surfactants can be done within such a group, and how this can be clearly assigned to certain group members. And we made use of it and put this into this graphic here. And you can see that dependent on the C chain length and the etoxylation degree, that again corresponds to the hydrophobicity, we can put in certain threshold values, or at least assign certain areas with high toxicity, moderate toxicity in the white area, and low to absent toxicity in the green area. So acute tox for aquatic depends both on the chain length and the degree of etoxylation. And we can bring these into different subgroups when applying the limit values for acute aquatic toxicity implemented in GHS. So, and while the etoxylates included in CESE, they cover the linear ones, the quasi-linear and branched ones, this is not really considered to affect this illustration here, because as mentioned, we are always dealing with mixtures. You may now see here clear defined borders. This is, of course, for illustration reason and for better understanding, but however, we're still dealing with mixtures when talking on polymers. Regarding toxicology, there's also a comprehensive database available on acute and long-term systemic tox, as well as local endpoints. You can see the publications here, they're all available. And with the upcoming ECETOC publication, you will have a nice appendix summarizing all this data. So there is a lot of data there. And we can also work a little bit on what drives hazard. So the background for grouping for tox by hazard similarity, we identified based on all the data that the major hazard potential presented for these polymers is characterized by local action and local irritation. And they're either metabolized to physiological or occurring metabolites like fatty acids or compounds of low toxicity. So that's where we are mostly with these substances dealing with toxicology. And being able to characterize the key hazard, we were also able to put this into a nice data matrix. So the most relevant ones we identified are for oral acute toxicity and eye irritation. And you can see here in blue the outline for eye irritation and in purple for oral toxicity to put this into this graphic. On the next slide, you can see that the data matrix shows a trend for linear and branched alkyl etoxylates, both for eye irritation and acute oral toxicity. This is much dependent on the C chain length and degree on etoxylation, which is widely concordant, and what we may have seen similarly for the for ecotox. So, and if you go now down here, that we can merge these both areas and find a common area for acute oral toxicity and for eye irritation. To put this into a hypothesis for grouping and read across, we have a guiding hypothesis put up into the green area here. So the magnitude of ecotox and the relevant hazard property as well as for toxicity, changes in a predictable manner, as demonstrated before. Which means that hazard similarity-based approach is suggested as a relevant to subgroup if they exhibit the same hazard classification. And thereby we can do read across within these subgroups to fill data gaps of the target substances. And finally, as we do have a continuum of effects, and this is a mixture, and this continuum proceeds over all the entire subgroups, we can conclude that all alkyl etoxylates are considered a single overarching group. This is based on similarity, first, on the common key feature, like the amphiphilic structure, second, the trend behavior of physical properties, third, similar mechanisms for the relevant hazard properties, as outlined. And the hydrophobic part strongly interacts with biological membranes, leading to non-polar narcosis mode of action. And we put in some considerations for the foundation to fill data gaps as outlined down here. So to summarize, the amended grouping scheme presented has proven useful for grouping of alkyl etoxylates. The description along the three criteria also allows the description of polymer groups and polymer identity, with criterion 1 on structure, criterion 2 on trend behavior of physical properties, and 3 on the relevant hazard properties with similar mechanism. So overall, by identifying the key components and a coherent hazard similarity hypothesis, alkyl etoxylates polymer group was built, which is fit for purpose. Thank you very much for listening, and I may now hand over to my colleague Philippe for the next presentation. And I may quickly introduce Philippe to you. Philippe's got a diploma in chemistry from the University of Paris Tech, and further did a PhD in physical chemistry. His professional career further proceeded at Sieber and Huntsman with various leading roles in R&D and technical service. And his current role is with Huntsman in REACH as Global Risk Manager. So happy to take over to you, Philippe, and hand over. Thank you very much.
Good afternoon. Thank you very much for the introduction. I'm going to talk about the one of the examples concerning the epoxy resin polymer group. The original idea with this case study on batch epoxy resins is that according to Article 138, it was planned to issue a report on the risk posed by polymers in comparison with other substances. This report has not been issued, and it is still a missing piece in the community endeavor to develop a suitable polymer legislation. That brings us to the commonality and differences between polymers and substances. We see our case study was a try to contribute by laying a bridge between the well-known world of substances, for instance, as registered under REACH, and the less well-known polymer universe. The batch epoxy chemistry obviously provides such an opportunity. It helps understanding what happens when transitioning from substances to polymers when derived from common chemistry. This case study also provides an example of the tiered three-layer grouping as presented just before. This approach developed by polymer task forces by ECETOC as well as CEFIC. So first, the grouping starts with a clear identification of the common chemical features, was said. This case study strengthened the three-layer grouping scheme as previously described with a polymer fraction approach and introduces the, sorry?
This is Heli. Could you switch to full screen mode, presentation mode, so that slides become bigger?
Is that okay like this?
Yeah, better. Thanks.
Thank you. We first grouped the, let's say, according to a clear identification of the common chemical features, and we strengthened this three-layer grouping scheme with a chemical fraction approach, and we introduced with this case study the concept of key common constituents. Second, the physical chemical properties will help to refining the group identity and define the boundaries. And third, of course, the tox and ECETOC assessment will substantiate the similarity of acid in the polymer group. So let us start with the chemical identification. These epoxy resin oligomers polymers are obtained by copolymerization of BPA, bisphenol A, and epichlorohydrin. These are both difunctional monomers which align their links to produce a kind of linear chain with end standing with terminal epoxide groups. The sequence in the bracket is resulting from bisphenol A and epichlorohydrin in their reacted forms and represents the repeat unit of that polymer. So every polymer is a collection of, let's say, such oligomers with varying values of N. If you react BPA with a strong excess of epichlorohydrin, you obtain what you can see here, batch prepolymer, which is the, let's say, the oligomer with N0, N equals 0. And this prepolymer can be used either as a substance on its own right to be used in various downstream user technologies, or it can be also used to be reacted again with BPA to produce the same kind of polymer. So we can produce basically this type of polymer in two ways, by two different processes, either by directly reacting BPA with BP or by reacting the prepolymer N0 with BPA. These two processes are linked to CAS identifiers, which are different, although the resulting material is the same, because the CAS identifier mostly relates to the process and not to the substance you get at the end. So we can see that, let's say, CAS identifiers are certainly useful for polymer identification, but they are not, let's say, enough to, they have their mutations and cannot characterize the polymer 100%. We can see as well that the batch prepolymer is a non-polymer molecule according to the polymer definition, and all other oligomers with N greater than 0 are all polymer molecules, so that a resin, a polymer, is basically a mixture of substance molecule and various oligomers with different molecular chains. So we are basically playing, let's say, in a field where we have in the same material a substance and various polymer molecules, and that lay the bridge between the world of substances and the world of polymers. So we cover with this chemistry the whole chemical space, and we lay really the bridge between the substance and polymers. Just too quick. So, no worry, this is just a jet formation chromatography spectrum. Here, the thing we have to keep in mind, when we are chemists and not chemists, these peaks stand for the various molecules, if you prefer, the various constituents of the material. The blue line here stands for the batch substance, which we referred to before, and those different colors, spectra stands for polymer, epoxy polymers, so batch polymers, if you prefer. The thing we have to keep in mind is that some peaks are common to all these substances, especially this one, which is batch N0, which is the, of course, the main component, main constituent of the batch substance resin, and this peak is still present in the various polymers based on batch as well. So comparing the structure of batch here n equals 0 with that of any batch oligomer, it appears that their structures are homothetic. Higher oligomers are simply a repetition of the same chemical sequence, simply longer molecules. We do not really expect any different contribution to the tox and ecotox properties from two different oligomers, especially in the high molecular range, which is considered as a matrix. Therefore, a generic description of these high molecular weight constituents is sufficient. It consists of the repeat unit definition, as we have just seen, the chemical structure, for instance, linear with terminal epoxide groups, and of course the molecular weight distribution, which makes the difference between two polymers. We assume batch N0 to be the constituent with to be the common tox driver, since it is present in all these polymers, and it is the constituent with the lowest molecular weight and the highest epoxide group ratio. And we assume as well the other oligomers to play a minor role in the story. So we finally consider batch N0, this stuff here, to be the key common constituent, and the batch substance, which contains, as we've seen, this constituent as main constituent, to be a suitable reference for comparison. So now, what do we consider to be a key common constituent? Here we have gathered the main features. It is present in every polymer of the group. This is a common chemical feature, if you prefer. Or reversely, a polymer group could be identified with a group of common key constituents. It has to be soluble in a GPC solvent for easiest and more accurate substance type analytical identification, so that you can make a GPC spectrum, for instance. It is well manageable with KSR tool, which is, let's say, well suited for molecules with molecular weight below 800. It has the highest probability to be physically available, to be soluble in water and fat, and also bioavailable for systemic effects via cell membrane permeability. It has the highest probability to drive the toxicity and ecotoxicity based on reactivity, the mobility, and accessibility of the reactive groups in small molecules. It has most likely a full data set available, or part data set available, as, for instance, registered pre-polymer or monomer, additive, etc. And you can see that it is a very powerful mean of, let's say, group identification and hazard assessment as the common active constituent. So this chart represents the correlated variations of the batch N0 constituent with the epoxide functional group concentration. So simply saying logically, when the molecular weight increases, the two others here, batch N0 and epoxide index, decrease in that direction. So that here we have low molecular weight domain and high molecular weight domain. Above 60% batch N0, we have the batch substance registration domain. Below, let's say 18%, batch N0, the resins are solid. In between, we have a domain of quite viscous resin with processability. They are not of practical interest, and they can be easily replaced by mixtures of batch substance with any of these high molecular substances, which are polymers. So the solid resins are delivered as dust low flakes, and the different technical grades are named types. Type 1 is a solid resin with the lowest molecular weight here, and the highest ratio of epoxide groups. There are obviously at the end only two domains which are really of interest. This is batch substance, which is already registered, and the domain of the solid polymer resins, starting from type 1 upwards. Consequently, the physical chemical properties help us here to define the lower boundary, which would be the type 1 resin. And the upper boundary would be an open-end one, since we know resins with molecular weight of 30,000 and above. However, the scope of our study is limited to those types with, let's say, some measurable residual batch and zero, like here in this domain. Okay. So once the key common constituent has been identified, how do we assess its physical availability first? This table provides the identical values, which substantiate the first layer group chemical grouping. All the resins contain the key common constituent, batch N0, to various extent. Here, of course, the resin, the batch resin, has a very high level of typically of batch N0, and this is decreasing then towards high molecular weight or higher type resins. So this is my note if I forget something. So by the way, you have here a collection of the GPC parameters, which help defining whether we fulfill the PLC or the PR criteria. Now, the main point is, how do we measure the physical availability? We do it via water extraction test in relatively severe conditions. So we grind first these solid resins and we let it steer for 20, 30 hours at 30 degrees. And what we find is that batch N equals 0 is detectable at ppm level in the type 1 and the type 2, which are the lower types, and the dimer is not detected. And of course, we assume that the higher oligomers wouldn't be detectable either.
Philippe, 2 more
Yes.
minutes.
Okay, so I think I need some more. So even if only at very low level, batch N0 appears to be the only physical available fraction from batch solid resin. And we do not expect a contribution from other oligomers. Therefore, we assume a similarity of acid among the batch oligomers, or about the batch polymers, with only differences in the intensity, which was called the effect level. It hence appears as justified to evaluate the solid resin with reference to the batch substance. To check the bioavailability of batch N0, we test solid resins for the endpoints, which are positive for the batch substance. Batch was registered as rich substance in a tonnage band 100,000 to 1 million. So the full data set is available, and the batch substance actually doesn't show a potential for systemic toxicity in mammals. But as indicated here in the table below, because skin and eye irritation, as well as allergy skin reaction, and it shows as well some chronic toxicity to aquatic organisms. Now, these are the test items. We take a type 1 and a type 2, as well as their purified versions, from which we have removed batch N0 by distillation. The results are collected in this table. So again, we have the definition of the 4 test items and the GPC parameters. And we focus now on the results in the lower part of the table. Batch substance is not very, let's say, water soluble, but the polymers, the solid resins, are insoluble by a limit of detection of 2 milligrams per liter. And the overall picture provided by the other test is that batch N equals 0 is not bioavailable, or not to an extent which would manifest the toxicity of batch N0 in the polymers. We, however, have two outliers provided by the purified resins, but these outliers results cannot be explained by, let's say, considerations on the concentration. So let me please summarize the main takeaway learnings from this case study. We have let's say, using the analytical and the chemical data, we can work out a first layer grouping proposal. Although batch polymers can be obtained via two processes, with two different cast identifiers, there is enough evidence to make one group. And cast identifiers are useful, but not sufficient to unambiguously identify polymer groups. The batch chemistry produces continuously varying composition from batch substance to a wide range of low and high polymers, thus providing a better understanding of the type of acid to be expected when moving from substances in general to polymers. The analytical data generated allow to assess a PLC-PR status, and we use a polymer fraction for identification and grouping. The low molecular fraction, as shown in GPC spectrum, present in every batch polymer to various extent. This low molecular fraction contains well-identified constituents, including the key common constituent, which is the main common tox driver. The high molecular fraction doesn't require identification of every oligomer. It can be conveniently well-defined with the repeat unit, the molecular weight distribution, the reactive functional groups. Polymers such as batch polymers are well-defined multi-constituent type polymers. As a second layer, the physical chemical properties come into play. For refined grouping or subgrouping. As a result, the lower boundary of the polymers in scope is defined as the low molecular type 1 batch solid solution. These are the physical chemical chemistry properties which help us for that purpose. The physical availability of batch N0 as key common constituent was measured upon water extraction test and proved to be extremely low, ppm. And at the third grouping layer, low-tier tests were performed for the endpoints, which are positive for batch substance. Hazard similarity supports selected grouping. Only differences in the intensity of acid are expected. Two resins were selected at the low boundary of the polymer range in scope for so-called bracket testing, with average molecular weight bracketing 1,000 Dalton. The two resins, both as produced and without batch, were tested. The results show overall no bioavailability of batch from the solid polymer resins in scope, but some inconsistency involving the purified versions suggest that the used test methods are not fully suitable for polymer and merit some adaptation work. With that, I'm done. Thank you very much. Sorry if I took a little bit more time as planned. And it's my privilege to introduce the next speaker, Isabel Cook, who obtained a diploma in food chemistry at the Department of Food Chemistry and Toxicology in Kaiserslautern in 2014. She worked, PhD work bore upon in vitro studies of the effects of mycotoxins and marine biotoxins in the blood-brain barrier, which she defended in 2018. Afterwards, she joined the ASF, where she is currently working as regulatory toxicologist. Thank you very much. Hand over the floor to Isabel, please.
Isabel, this is Heli. Could you please try to stay within your 15 minutes sharp, or even speed up a bit more, because we're
I will give my best. Can you see my presentation modus?
Yes, that works. Thanks.
All right. Thank you so much for the kind introduction, Philippe. And today I want to talk about the grouping of polymers and read across to corresponding oligomers with the example of the polyethyrols and short petioles. So first, I want to start off with an introduction of the polyetherols. They are polymers based on initiator molecules which contain multiple hydroxyl or amino-functional groups. These initiator molecules are rather simple molecules like glycerol, diethylene glycol, or more complex molecules like sugars, sugar alcohols, or also linear or aromatic amines. But today the focus is not on the amino-linked polyols, but rather on the ether-linked ones. So these initiator molecules are then alkoxylated either with ethylene oxide or propylene oxide or both to varying degrees. And the result is polyetherols with varying chain lengths, which are fulfilling the polymer criteria. Polyols are mainly handled in a controlled industrial setting, and they are further processed downstream. For example, they can undergo reactions with diisocyanates to produce foams or mattresses, or for mattresses or insulation boards, for example. Polyols are also mainly used to form other polymers, but this is not the focus of this case study today. So in general, no consumer or professional use is anticipated, and the exposure or wide dispersive use is therefore considered unlikely. Like I just described in words, here you can see an overview of the polyols included in this case study. On the left, you can see the structures of the initiator molecules, and in the right column, the alkoxylation variants. On the right side, there's a reaction mechanism for the alkoxylation. So they can either be ethoxylated, propoxylated, or both ethoxylated and propoxylated. So the R1 represents the initiator molecule, which is then reacted with the alkoxylation. So either ethylene oxide or propylene oxide. And depending on the desired application or use of the polyol, different degrees of alkoxylation are achieved. Which also results in a range of constituents of different alkoxyl chain lengths, which are found within one polyol. On this slide, we go further into the grouping of the polyols. The basis of the grouping of the polyols lies in their chemical nature, physicochemical properties, and mainly the hazard similarity. Also, their application domain is very similar throughout the polyols. Some basic acute and local toxicity data are already available for selected polyols, and for those where there are some data gaps, they can be filled by the use of read-across to corresponding so-called no longer polymer polyols, in short, NLPs. For this, the NLPs with the same or similar initiator molecule serves as an appropriate data source for the read-across. I will explain this shortly. So the hypothesis for the grouping can be described as follows. The polyols of this case study are grouped in one group based on their properties and the hazard similarity, with potential subgrouping by the initiator molecule. And a general theme is that with increasing alkoxylation, the influence of the initiator molecule decreases, which is also generally accepted concept for systemic bioavailability, that with increasing molecular weight, the bioavailability decreases. This means if no or only minor effects are seen for the smaller, more bioavailable NLP polyols, the systemic effects for the larger polymeric polyols are not expected to be higher than those seen for the NLP polyols. So when we think about how or why it is appropriate to use the NLP polyols as a read-across source to assess the polyols, we can think of the NLP polyols as the smaller counterparts of the polyols. In principle, the NLP polyols, as well as the polyetherols, so the polyols, are based on the same principle. The same initiator molecules are alkoxylated to produce either NLP polyols or polyols. The only difference here is the degree of alkoxylation, meaning different chain lengths, resulting in different molecular sizes. So whether we are talking about NLP polyols or about polyols is purely a regulatory cutoff, whether it is regarded as a no longer polymer or as a polymer. This is illustrated in this schematic GPC, which helps to explain the difference. Here on the left, you can see that the majority of constituents in the oligomeric polyols does not fulfill the criteria to be regarded as polymers. And on the right, you can see that the majority of constituents of polyols is of higher chain length. But it is all a distribution, so there is still an intersection of constituents which are both available in the NLPs and the polyols, with the same initiator and alkoxylation. So let's have a look at the data situation. The physicochemical data are available for those substances. However, for the environmental data, there are very little data available for polymers, but for the NLP polyols, for the smaller brothers, the basic data sets are available. For the human health part, there are some acute and local toxicity endpoints are covered for selected polymers for the polyols, and a basic data set also including limited data on repeated dose are available. So I'll jump into the data for the physicochemical properties to set the first criteria, or no, it was, yeah, the chemical nature was the first one. The second one is the physicochemical properties. So we have a molecular weight range from higher than 500 Dalton up to the highest around 18,000 Dalton. They are highly viscous liquids with a good water solubility, and the log KOW is below 1. Only for a few polyols it is ranging between 1 and 2, and in general they are not surface active, and also they show a low vapor pressure. So the physicochemical data support the boundary description of the polyols as a group, because they are pretty consistent within this group. For the environmental properties, robust and publicly available data are scarce regarding the environmental phase data. But for NLP polyols, there are several endpoints are covered, and they are mostly readily biodegradable. And also for the NLP polyols, it was shown that no acute aquatic toxicity up to 100 milligram per liter was observed, even though the NLP polyols are highly water soluble. and in the few chronic studies, also low aquatic toxicity was confirmed. Therefore, no concern for bioaccumulation was identified. Again, our hypothesis also applies here, namely that it is not expected that the corresponding pools, which are based on the same initiator molecules and alkoxylation, but have a higher molecular weight, exhibit more pronounced toxicity than their lower molecular weight and shorter chained NLP counterparts. The data for the NLP counterparts did not raise any concerns, and the few studies that we do have for the pools support this hypothesis. So in summary, we conclude that no environmental hazard for pools is expected. So for now, we come to the next part, namely the human health properties. Also here, the hypothesis for the grouping is the same as for the environmental tox part, meaning that it is not expected that the pools have a higher toxicity than the lower molecular weight and shorter chained NLP polyols counterparts. This is based on the fact that the bigger a molecule becomes, the lower the systemic bioavailability will be. So with molecular weights of pools being higher than 500 Dalton, the diffusion in the gastrointestinal tract is expected to be limited and only possible for some of the smaller constituents within the substance. But always keep in mind that systemic bioavailability per se does not automatically translate into systemic toxicity. So even though the NLP polyols and lower molecular weight pools have the potential to become systemically bioavailable and reach cellular targets in the organism, this does not automatically mean that there is any toxicity observed. So for the NLP polyols, no local and acute systemic toxicity was observed. Also, limited toxicological data for repeated dose toxicity indicate low systemic toxicity, in most cases even up to the limit dose. For the pools themselves, low to absent local acute toxicity. For the endpoints of acute dermal toxicity, skin and eye irritation, mutagenicity in bacteria, and skin sensitization was reported. So in general, no acute, and also in general, no acute oral and acute pulmonary toxicity was seen for the majority of tested pools. But I have to mention here that limited data indicated slightly more pronounced acute oral and or inhalation toxicity for glycerol or propylene glycol initiated pools in a certain molecular weight range. However, more further research is needed here to fully understand this exception. On basis of the information we do have at the moment, it can be said that the pools show low to absent local and acute toxicity. No inherent potential for to induce systemic toxicity is assumed. And then I already come to my summary. I hope this still fits in the time.
Yeah, 1 minute is good.
The pools are handled mainly in an industrial setting and are used as pre-polymers to further be processed downstream. They have as a common key feature, they generally possess a low hazard potential for the environment as well as for the human health. They are grouped as one group within the pools and also make use of the read across to structurally related smaller counterparts within the NLP polyols, which serve as an appropriate source to cover certain data gaps. So the common features are found in their chemical nature. their physicochemical properties and the low hazard similarity. So based on the information we do have, no hazard has been identified for the NLP polyols. Therefore, no hazard is assumed for the bigger POs.